Hi, everyone, and thank you so much, Peyton and Renad and everyone else from Bioprotocol. Let me get my screen shared here. Okay, can everyone see and hear? Okay, great. Yeah, thank you all for tuning in. I am really thrilled to tell you all about some of the work going on in the Blue Lab right now to develop and enhance prime editing for experiments in mammalian cells in this case, but they have many other applications too. So feel free to, if any questions come to you throughout the talk, feel free to go ahead and put them in the chat. And yeah, we can just get started. Okay, so just to kind of give you all an idea of what we'll be going over today, I think it's important to talk about when we should even be using prime editing relative to other genome editing technologies, what are kind of its optimal use cases. We'll discuss how prime editing works, and then we'll spend most of the time today on this third point, which is how you should design and optimize a prime editing strategy for a brand new target of interest for your own project and your own applications. We'll kind of finish off by doing some example applications. I'm really excited to tell you about two of my coworkers' recent publications just within the past month that use prime editing for some really interesting therapeutic applications, and then we'll have plenty of time for questions at the end, so feel free to use the chat. So just to dive right in, if you're attending this webinar, you're probably interested in genome editing. And at this point in time, there are it's a really complete and robust toolbox that we have for genome editing in many different organisms. Kind of the first iteration of this and really big breakthrough was Cas9 nuclease. And traditionally, when people use Cas9 nuclease, you're targeting the Cas9 protein to induce a double-stranded DNA break at a target site in cells. And if you just do this, you'll generate indels or insertions and deletions at the target locus. And if you add a DNA donor, you can do HDR or homology-directed repair to install specific mutations. Other tools developed later on from the Lu Lab are base editing, which can convert specific bases to other bases, so C to T or A to G, and we refer to these as transition mutations. Unlike Cas9 nuclease, these do not go through a double-stranded break intermediate, which is really useful, but again, they can only do these transition mutations. And the subject of today's talk is going to be prime editing, in which you use a reverse transcriptase fused to a Cas9 negase to kind of synthesize this DNA flap intermediate. And you can use it to install any small DNA change, and you can use some kind of improved and enhanced versions of prime editing to make even larger changes to the genome. And, Cas9 nuclease, base editing, and prime editing, they all have their pros and cons, right? If you just wanted to knock out a gene, for example, you might just want to use Cas9 nuclease. It's efficient, it's fairly easy to do, but the downside is that it does only generate indels, and you often can't control what these indels are. Let's say you wanted to install, a specific mutation in an HDR competent cell type, then maybe you would use HDR. It can be really versatile and very efficient in the cell types that it works in, but unfortunately, it doesn't work in all cell types, such as postmitotic cells. And because you're still going through a double-stranded DNA break, it can lead to other undesired consequences like large deletions, indels at the target site, large translocations, other undesired byproducts such as that. Base editing, like I mentioned, is very efficient and they're very, very good at what they do, but they're only limited to transition mutations, so C to T and A to G changes. And that kind of leads us to prime editing, which in a lot of ways is kind of a lot of the good qualities of all three of these technologies wrapped into one. So it's versatile. You can install any mutation that you want as long as it's small. And like I said, there are ways to manipulate prime editing for larger changes. And if you optimize your prime editing approach well, you can have very low indels and very efficient levels of editing. And it's been shown to work in a wide variety of cell types. So primary cortical neurons isolated from mice. It works in vivo in the CNS. It works in the liver, in plant cells, E. coli. So it's really versatile. It's really flexible and it can be really efficient. So it's a really powerful technology. But the downside of prime editing is that it requires a bit more optimization and a bit more time spent on your editing technique than some of these other technologies that I just talked about. But that's kind of what we're here to address today. So how does prime editing work? So I mentioned that prime editing uses a prime editor protein, and it also uses this modified CRISPR guide RNA called a PEG RNA. A prime editor protein is made of a Cas9 nickase fused to a reverse transcriptase, and the pegRNA that it uses is in some ways very similar to a normal CRISPR guide RNA. So it has this scaffold sequence that can bind to the Cas9, and it also has a spacer, just like normal guide RNAs, that targets it to a certain spot in the genome. What it has that most CRISPR technologies don't use is this 3' pegRNA extension, and we'll get into what exactly is in there in a minute. But let's say that you've managed to deliver your prime editor and your pegRNA into a cell to do genome editing. The two associate, and then the guide RNA is able to target the prime editor protein to a specific spot in the genome. Because you're using a Cas9 nickase instead of a nuclease, it will nick just one strand, so just this top strand of DNA, and that frees this 3' flap of genomic DNA. And this is where that 3' extension that I mentioned comes in. So what you have now is you have this genomic flap that you've generated from your nick, and you can design this 3' extension to have basically any sequence you want, and we kind of split it into two parts. So the very end is what we call the PBS, or primer binding site, and then immediately after that is the RT template. So what do we design these things to do? So the PBS is designed to be complementary to this genomic flap that you've generated. So it base pairs with the DNA from the genomic flap, and you create this DNA-RNA duplex from the PBS in the genome. This is then recognized as a substrate from your reverse transcriptase enzyme that is fused to the Cas9 nickase. And the enzyme will start reverse transcribing using the genome as a primer, and it will add bases onto it based on what is encoded in the RT template. So you're able to kind of dictate whatever you want to put into the genome based on the sequence of your RT template. So for example, you might encode maybe 2 bases that you want to edit by encoding those changes in the RT template. The RT will synthesize those bases onto the end of your genomic DNA flap. And it's also very important to include downstream homology in your RT template as well. And that's because when the reverse transcriptase is done with its job and it finishes synthesizing the RTT templated bases, this complex will fall off and you'll be left with a flap with your edit, but also this downstream homology. The downstream homology allows the flap with your edit to kind of lay down and kneel into the genome. And then you have what we call flap equilibration. And in a series of DNA repair steps that we kind of partially but don't fully understand, this intermediate is either resolved as the edit being permanently installed, or it can also be reverted back to the wild type sequence. So this is kind of the general cascade of prime editing events for what we call the PE2 system. So to kind of update everyone on some nomenclature, PE1 is the original prototype prime editor where members of our lab just kind of stuck together the MMLV reverse transcriptase and Cas9 nickase. PE2 is a huge improvement over PE1, and it uses mutations in the MMLV RT that enhance its activity. So basically, there's no reason to use PE1 anymore. PE2 is kind of our baseline prime editing system, and it's useful for a lot of applications. However, we kind of run into trouble with these arrows here. So this intermediate can be either repaired as we wanted to with the edited sequence being permanently installed, or it can be reverted back to the wild type sequence based on this bottom strand here. And this is dictated by cellular MMR. So there are a few different advances to prime editing that have allowed us to kind of enhance this arrow here towards the edited sequence and away from reversion to the wild type sequence. One thing you can do is you can just add another guide RNA. So it's not a pegRNA, it's just a normal CRISPR guide RNA. And this, because it's, there's no 3' extension, the only job of this second guide RNA is just to direct the prime editor protein to nick this opposite strand. Because mammalian mismatch repair is nick directed, the addition of this nick kind of confuses the cellular MMR machinery, and instead of reverting things back to wild type, it becomes more likely to install your edited sequence. Another way that we can kind of bias these MMR outcomes in favor of the edited allele is by adding the MLH1DN, which is a dominant negative mutant of the protein from the MMR pathway MLH1. So this inhibits cellular mismatch repair, and much like the nick that we just talked about, kind of shunts these intermediate products towards the edited outcome. So when we have just a nicking guide, we call that PE3. When we have just MLH1DN, we call that PE4, and when we have both of them, we call that PE5. So this is kind of the landscape of prime editing systems that are available to us. So how do we use them? There are, There are so many different things that you can do with Prime, but basically no matter what your application is, you're going to start with these 5 steps that are common to basically any Prime editing project. You'll start off by selecting your target edit. You'll find protospacers that can target that edit. You'll optimize your PBS and RT template lengths, find nicking guides, and then select one of the PE systems that we just talked about. Something I haven't mentioned, but we'll get there, is the addition of silent edits. And between these 5 things, you can really, if you kind of carefully go through them all, you can really get efficient Prime editing out, especially if you're willing to kind of iterate and optimize your editing approach. By doing this, you'll have, a way to hopefully very efficiently style your edit of choice. And then there are many different ways Prime editors have already been used. The 2 that we're going to go over today is RNA delivery for ex vivo editing of therapeutically relevant cells. And then we're also going to discuss dual AAV delivery of Prime editors. But like I said, Prime editors have been used in many different organisms. So, if you have an application in mind, it's probably at least worth thinking about. So just to kind of dive right in with the first thing we might want to do, step 1 is to select a target protospacer. So let's say that you've identified the base that you want to change, and you'll start off by scanning the locus for PAMs. So much like all these other Cas9 derived tools, Prime editors use SPCas9, and it requires a PAM or a protospacer adjacent motif, which is made up of NGG, so N being any base and then 2 Gs. So you'll identify PAMs that are near your target base, and the PAMs will dictate a corresponding protospacer, so the 20 base pairs immediately preceding the PAM. We tend to prioritize target protospacers that are very close to the target edit. This is not a perfect assumption, but you definitely don't want to be using protospacers that are exceptionally far away. So we'll prioritize the most proximal protospacer. And the only other thing you really need to consider when selecting a protospacer is that if there's one that would necessitate you to include a long poly-T sequence in the plasmid encoding your pegRNA, you should avoid that because those poly-T sequences can prematurely truncate the expression of RNAs from the U6 promoter. So these are kind of the 2 things to keep in mind. You want to be close to your target base and you want to avoid poly-T stretches. If we zoom into this target protospacer 2, we can start to design the rest of the pegRNA. So like I mentioned, there's a PAM here, this TGG, and this dictates what the protospacer is, the 20 base pairs before the PAM. Cas9 nicks between positions 17 and 18 of the protospacer, so the nick will occur right here. And the only bases that are Prime editable are the ones downstream of the nick, right? Because we're going to be adding bases onto this top strand after the nick. So for example, if your target edit was here, you wouldn't be able to access it by Prime editing with this protospacer in PAM. But because our desired edit is over here, so plus 11, meaning 11 bases away from the nick, let's say that we wanted to change this G to a T. What are the other components of the pegRNA that we would have to design in order to do this? So just to kind of keep everyone on the same page, this is what a state-of-the-art pegRNA looks like. You have this spacer, and this corresponds to the same sequence as the protospacer shown in the DNA here. So this will target the editor to the target locus. This scaffold is consistent for most SPCas9 applications. It's just what the Cas9 binds to recognize the guide RNA. So that is basically constant no matter what you're doing. The other constant part of the pegRNA that we haven't talked about is this TiVo pre-Q1 motif. So it stands for trimmed Evo pre-Q1, and it's just a highly structured motif that is used to stabilize the pegRNA, prevent its degradation, and this really improves Prime editing efficiencies quite a bit across the board. And this was actually an advance that was co-invented by Peyton, so we'll direct any questions about this to him later on. So beyond these kind of constant scaffold and TiVo pre-Q1 motifs, in addition to the spacer that we've already talked about, the 2 things that we need to address are the PBS and RTT. And the main ways that we change these are by their lengths. So you can imagine that we could extend this PBS to have either, more homology to the nicked flap that forms, or kind of be shorter and be complementary to less of the flap. For the RTT, we know it's always going to start at the nick because that's where the RT will start reverse transcribing. We know it's always going to contain the target edit. So for us, we want to change this G to a T. So we'll template that with an A in the pegRNA. but we don't know how long to extend it for. So how much downstream homology do we want the RT template to encode? So these are kind of the two lengths that we play with. How much hybridization do we want the PBS to have, and how much downstream homology do we want the RTT to encode? To give you a ballpark of the lengths that we typically use for these, almost all PBSs, it's pretty easy. They'll usually fall between 8 and 15 base pairs. So we'll usually select maybe 3 within this window to start out with. And this is pretty agnostic to the edit type. It's kind of always in this range. The RTT is a little bit more complicated because it depends on the edit that you're encoding. So you definitely want to have downstream homology no matter what the edit is. But kind of what we typically do in the lab is if we're doing a small change, like the single base pair change, you don't need so much downstream homology, maybe, 10 or so bases. We're kind of varying the total RTT length between 18 and 24 for this edit. But if you had, for example, a longer edit, maybe you're inserting a flag tag like people have done in the original prime editing paper, you would want more downstream homology because of the larger change. So that's one thing to keep in mind. And then the other concern for the RTT length is that you don't want it to be at a length such that the first base of it is a C. We've found that that can disrupt interactions with the guide RNA scaffold. So that's just kind of a pitfall to avoid. So these are the things that we consider, and typically when we're designing a new prime editing strategy in our lab, we'll pick a couple PBS lengths, a couple RT template lengths, and then the number of pegRNAs that we test is just kind of a matrix of those. So if you were to design 3 and 3, it would be 9 pegRNAs for kind of an initial first pass screen. So that's everything that we needed to cover for the pegRNA. But you also will probably want to be, and remember to always include the EPEG modification because it's really helpful for editing. So in addition to the pegRNA, you'll probably want to design nicking guide RNAs because they're really also very helpful for boosting editing efficiency. We want to be nicking the opposite strand relative to where the prime editing is happening. So if your PAM and ePegRNA spacer are on the top strand, you'll want to look for PAMs on the bottom strand for your nicking guide. The other consideration for the nicking guide is that typically, although not always, these nicking guide locations in the plus direction or kind of on this side of the nick tend to perform better than nicking guides that target the minus direction, such as over here. So for example, you might want to prioritize these nicking guides if you have several options. The other consideration for nicking guides is that if you get very lucky, you might be able to do what we call a PE3B strategy. So the PE3B strategy requires that you have a PAM that happens to be on the opposite strand and close to the site that you're trying to edit. So for example, if you're trying to do a +1 T to G mutation here, you'll notice that there's a PAM on the bottom strand. And what PE3B is, what this allows you to do, is it allows you to design your nicking guide so that it can only target that top strand after the RT has already synthesized your mutant flap. So for example, if we're using this bottom PAM, we'll have this nicking guide protospacer here, and you'll see that this C here does not perfectly match with the T that is in the original wild type allele, but it does match perfectly with the G, which is the edit that you want to encode. So this nicking guide is only perfectly matched after the first several steps of prime editing have happened, which is really beneficial because this can decrease the level of indels that you see. And we've used PE3B strategies to really improve editing to indel ratios at a few different sites in our lab. So those are kind of the main considerations for the nicking guide. In many ways, it's much simpler than the pegRNA. You're really just finding protospacers. There's no need to design an EPEG motif or a PBS or an RTT or anything like that. And one other note about PE3B is that I think everyone I know who has tried to design a PE3B strategy at one point has thought that they could do this with the PAM itself. But you cannot do this with the PAM, only with protospacer bases, because what you're recognizing is the other strand, whereas the PAM is on this bottom strand here. That's just like a really common error that a lot of people make, myself included. So watch out for that. Okay. One of the last things that you'll need to think about, and I haven't really mentioned this before, is the addition of silent mutations. So just to kind of contextualize when these are useful, let's go back to our original edit, this plus 11 G to T. Let's say you found an optimal PBS, you found an optimal RTT, you do prime editing, and this is kind of that same intermediate we talked about with the mismatch. This is well on its way to being edited, but there are a few problems with this intermediate. Problem number one is that this TC mismatch forms a kind of mismatched DNA bubble that is very efficiently recognized by cellular mismatch repair. So, we've talked about competing back to the wild type allele versus the edited allele. So this type of mismatch, kind of the shape of this distorted DNA helix, is going to be a very good substrate for reversion back to the wild type allele. The other problem with this intermediate is that even if, let's say that you do resolve this mismatch the way you want and your edit is installed on both strands, you still have a PAM intact here, which means that the prime editor can come back, nick again, reverse transcribe again, and potentially mess up your edited product. We can get around both of these problems by installing silent mutations in the RTT in the same way that we install the desired edit. So you want to make sure that these don't change amino acids or anything else significant about your site, but it can be very helpful to, say, have an RTT like this one with these silent mutations with this G and this C encoded in the pegRNA here. And if you use this kind of silent mutation containing RTT instead of the original one, your intermediate will look a little bit more like this. And this is beneficial for a couple reasons. So one, this bubble is now not just a single base mismatch. It's now kind of this larger distortion of the DNA helix, which counterintuitively is actually recognized less strongly by cellular mismatch repair machinery. So this is more likely to evade MMR proteins that would like to revert your edit back to the wild type allele. Secondly, this PAM, or this TCG sequence, is no longer a PAM, right? The PAM has been mutated, so after editing, you can't have the prime editor protein come back, re-nick, and start the whole process over again. So these two modifications really help this intermediate get converted to an edited outcome and stay there after editing is completed. So that's how we use silent mutations to enhance prime editing efficiencies. And then finally, we've kind of already touched on this, but I think it's worth considering more from an experimental design perspective, is that there are 5 prime editing systems, PE1 through 5. And, each one kind of lends itself to certain applications, but not all applications. The only one that you'll never want to use is PE1. Like I said, this was a really useful prototype. It was absolutely necessary for the invention of prime editing, but it's never going to be the most efficient thing to use. Kind of the baseline that you would want to start out with is PE2, and this is just the Cas9 RT pegRNA system. There's no nicking guide, there's no MLH1DN. Honestly, this is probably never going to be the most efficient approach, but it is nice in that it's the simplest. So let's say that you're just screening pegRNAs and you don't want to have to worry about optimizing a nicking guide just yet. You don't really want to mess with MLH1DN, inhibiting cellular mismatch repair. PE2 might be a good place to start. But if you really want to get good editing efficiencies, what I would recommend is the PE3 or PE5 systems. So both of these use an additional nicking guide that can boost editing efficiency, and PE5 also uses MLH1DN. And choosing between these two systems is going to depend on a few different things. The first one is going to be your edit type. So like we mentioned in the silent edit slide, MLH1DN, or mismatch repair in general, really recognizes these single mismatch mutations. So if your target edit is a SNP, it's very likely that MLH1DN will help. If you're doing a longer insertion or a longer deletion, it's less likely that MLH1DN will help. I think our cutoff is like 4 to 5 base pairs usually. So that's one way you can choose between PE3 and PE5. If you have a lot of indels in the PE3 system, you might want to switch to PE5 because PE5 has been shown to minimize indels. But on the other hand, if you don't want to inhibit cellular mismatch repair globally, you might want to stick with PE3. So I think of PE3 and PE5 as kind of similar, but certain aspects of your application or your edit type might tip the scales one way or the other. And then PE4 is kind of an intermediate. So it just uses MLH1DN, but no nicking guide. It's useful because it probably has the highest editing to indel ratio of all of these systems. So if you have an especially indel prone site, it could be very useful. But I think its biggest help is really for screening pegRNAs, because much like PE2, you don't need to worry about, optimizing a nicking guide RNA at the same time. But, this is probably going to be more efficient than PE2. So those are kind of the scenarios where you might choose one PE system over the other, or sometimes, we might test out two of them for a certain edit. It just kind of depends on how much you're willing to optimize. The nice thing is that there are certain things that you should always use. Like I've mentioned, you should always use an ePEG-RNA. They really improve editing efficiency. And the PE Max architecture, which is just you can use in any of these prime editing systems. So you might hear me say PE3 Max, for example. That just means that the prime editor protein is codon optimized and uses optimized linkers, optimized NLS sequences, and a few Cas9 mutations. So you should always use those two things to enhance editing efficiency. So at this point, you might be thinking to yourself, okay, let's say I have managed to choose just one protospacer. I now have 9 PBS RTT combinations, maybe 3 nicking guides. Maybe I want to choose a couple different silent edits and maybe try with and without MLH1DN. This can kind of become a very overwhelming matrix very quickly. But I think if you just kind of do it kind of layer by layer, and I'll talk about how our lab does this, it's actually possible to optimize all of these things without using a huge amount of resources or time or sanity for that matter. So typically what our lab does is that no matter what we plan on doing with the prime editing approach, we always start in a very easy to work with workhorse cell line that we've engineered to have the exact target edit that we want to correct. So if we're doing a human genome target, we'll use HEC293 T cells. And if we're hoping to do a mouse model of something, we'll work in N2A cells. It's really helpful to have a workhorse cell line that's easy to transfect because you're going to be screening a lot of different combinations. So sometimes we'll use prime editing itself, or we'll use HDR or base editing, some technique to install, maybe a pathogenic edit, and then we'll make a clonal cell line in HEC293T or N2A cells of that edit. So then we have this really useful platform to do the screens in. We'll then use Golden Gate cloning or Gibson cloning or some other method to clone a small initial library. I hesitate to say even library because it's only about 9 to 12 plasmids initially of pegRNAs. In terms of an initial first pass experiment, there are kind of 2 different ways our lab does things. So one thing we might do, like I mentioned, is we'll say we don't want to worry about nicking guides right off the bat. That's just going to confuse things and lead to too many samples. So we'll do either PE2 or PE4, so with or without MLH1DN. And the only thing we'll optimize is those PBS and RT template lengths. So that's one kind of first pass experiment we might do. The other type of first pass experiment we might do is say, okay, we'll optimize PBS and RTT lengths, but we'll also screen a couple of nicking guides. And both of these approaches are a reasonable number of samples. So the top one is typically, like I said, 9 to 12 samples per replicate. And the bottom approach is, if you multiply that by 3 for each nicking guide, like 27 to 36. So if you're doing this in an easy to work with transfectable cell line in a 96-well plate, which is what our lab does, it ends up being a reasonable number of things to screen. So after that first pass experiment, what you'll do, so you'll have these pegRNAs, each one will be transfected into its own well of a 96-well plate in this nice cell line that you've made. Maybe you're going to use MLH1DN because you don't want to optimize nicking guides. So you'll have your first pass experiment, you'll do a transfection, and then you'll perform HTS or whatever readout that you're going to do 3 days later. And you'll see where you initially land with your editing efficiency. And this kind of first pass outcome can be very different between different types of edits and different sites. So once you kind of see what your starting point is, it's then kind of up to you the number of rounds of iteration that you want to do to improve. And, if you're just using prime editing to kind of install a mutation and then you can screen clones, maybe you really don't need the editing to be that efficient. If it's a therapeutics project or you want to do in vivo editing in a mouse, you'll probably want to get the editing efficiency as high as you can. So then you can start iterating. So first of all, maybe you see patterns based on your PBS and RT template lengths. Maybe the longest RT template length you tried gave the best editing. So you might want to kind of go back and test even longer ones out. If you become happy with the RTT and PBS lengths that you screen, you can then start layering in these other improvements that we've talked about. So adding nicking guides, adding MMR evading mutations, adding PAM mutations, and then kind of using MLH1DN, testing these things in combination. So it's really all about how many things you're willing to test and, do you want to do it iteratively to save resources, or do you want to do it all in parallel to save time? This is just kind of an example of a campaign done in our lab. So these are for 2 different edits. The top one is for CXCR4 in orange, and the bottom is for this IL-2RB edit shown in blue. And this was the results of an initial PBS and RTT screen in HEC293T cell. HEC293T cells. And what's kind of cool is that you can see from this data that, there are different PBS and RTT preferences between edits. So this top edit here, it doesn't really seem like the PBS length is so sensitive, but for this one, you really only have one combination that's doing much better than your other lengths. So this really does show that you just have to test a few things if you're doing a new edit. You can then select your best one. So for example, the 17.3% here and this 4.72% here, and start layering in these other improvements. So this is just showing, I think, really beautifully how different prime editing improvements can stack together and take you from something that's really not usable to something that's pretty robust and something you might actually want to use in a project. So for example, this top one, it's that same edit from before. If you're doing PE2, you're only getting, maybe 2% editing. When you start to use the PE max architecture that you should always use, and ePEG RNAs that you should always use, and add in MLH1DN to get to PE4, you go from sub 5% to about 15%. And then if you layer on a nicking guide RNA with that, you can boost editing efficiency even further. So I think this data really demonstrates that it's worth kind of going beyond that first pass experiment, doing maybe 2 or 3 rounds of optimization to really get large improvements in editing efficiency. This blue graph on the bottom, this IL-2RB edit, I think shows something else that's important to consider when you're optimizing a prime editing approach. And that's that all of these different improvements can kind of interact with each other. So if you look at these 2 bars here, we're not using any silent MMR evading edits, and your improvement from PE3 to PE5 with the addition of MLH1DN is really large. So you're maybe doubling your editing efficiency when you add MLH1DN. But when you include these MMR evading edits, the PE3 editing efficiency increases a lot. And that kind of makes sense, right? Because both silent edits that kind of distort the helix and MLH1DN are doing the same thing, and that they're helping your edit evade MMR. So it kind of makes sense that the relative differences between these 2 bars would be different depending on the silent mutations that you're using. So you'll go through many rounds of iteration. You'll go through many rounds of iteration, and it does really pay off. But if this is all kind of overwhelming to you, if you don't want to spend weeks optimizing your prime editing approach, I do want to point out that there has been work from 3 other labs that is really interesting and exciting using machine learning and libraries of prime editors and prime editing efficiencies to predict which pegRNA you should use for a new edit. So Henry Kim's lab has 2 papers on this. Leopold Partz's lab has 1 paper on this, shown here, and then Gerald Schwank's lab has a paper on this as well. So all 3 of them have associated web tools where you can go to and you can plug in your wild type sequence and your desired edit, and it will kind of use this machine learning model that they've built to give you an estimate of the best pegRNA or the best 5 or 10 pegRNAs to use. The only one that we've been able to, the only one that we've tried to validate in our hands was the one from Gerald Schwank's lab, and that does produce pretty decent editing efficiencies right off the bat. I think we're excited to try out the other 2, but we just haven't gotten around to it yet. So these are really exciting. And if you found the first chunk of this talk to be overwhelming or intimidating, then this is a great place to start. And they'll definitely give you something to work with. Another kind of theme of tools from other labs that we use all the time for prime editing is that when we analyze prime editing outcomes, we use the Crispresso2 software from the Pinello lab. And I'm not going to go into too many details for this because a lot of it is just the same as you might use for another genome editing approach. But there are a few things that we use for prime editing that I think are pretty important to remember. So first is this discard indel reads, setting it to true. What this does is this throws out indel containing reads. So you're not counting anything with an indel in it as an edit. And it's also a very easy way to keep track of your indels because you can just count the discarded reads. Another thing that's important is this quantification window, this QWC value. And what this dictates is where you'll be looking for indels, where the program will be looking for indels. So, for prime editing, there are two different places where we can really see indels coming in. So one is where the pegRNA nicks. So for example, around here. And then the other, if you're using PE3 or PE5, will be around here. So there are kind of two places where you can see indels cropping up. And it's really important to include this entire range in your kind of indel window. So the QWC value you can define as 10 bases before your first nick, covering the whole region until 10 base pairs after your second nick. And that allows you to accurately and completely quantify your indels. So at this point, we've kind of gone through the whole prime editing workflow. I just want to touch on some controls, both positive and negative, that are going to be really important and really informative to include. The first one, by far the most important, is an untreated control, which sounds obvious, but if you're working with a very small number of cells to start with, it's very easy to get contamination from the environment or from you. And if you're working with a patient-derived cell line, it's not always clear what their starting genotype is. So the untreated control is really important for making sure that any desired or undesired things that you see in your sequencing output are from the prime editor. Another important control, if you're doing prime editing for the first time, it's probably going to be helpful to include a really well-characterized edit in addition to whatever edit you're trying to optimize. So for example, doing just a single base change at the HEC3 locus using one of the many ePEG-RNAs that we've published at this site, and making sure that you can hit the values that our lab has published is going to be really helpful just to make sure that as you're building a prime editing workflow in your lab that you're able to do everything well. If you're kind of testing a new cell line, for example, a cell line that prime editing hasn't been reported in before, and you're maybe electroporating mRNA or something, it's helpful to test those same reagents in a cell line that we know supports prime editing, just as a QC check on your RNA. And then finally, just kind of a related note, prime editing efficiency is really affected by transfection efficiency when you're working with HEC293Ts or N2As. So if you're comparing conditions, it's good to do that in the same experiment side by side to account for any kind of transfection differences in day-to-day or biological replicate to biological replicate. Okay, so that is pretty quickly how to optimize and kind of perform a prime editing experiment in mammalian cells. And we've kind of gotten to this point here where you've optimized your editing approach. And I just want to hit on really quickly two applications from our lab that have applied an optimized prime editing approach either with RNA delivery or with AAV delivery. So this first example was led by Kelsey Everett from our lab in collaboration with St. Jude. And what she did was she optimized a prime editing approach for correction of the sickle cell disease mutation. And then she delivered the prime editing components as mRNA. So she used a synthetic pegRNA and nicking guide RNA from IDT and Synthego. And she herself did in vitro transcription to make PEmax mRNA. She and her collaborators electroporated all of this prime editing machinery into patient HSCs from sickle cell patients, and then they transplanted the edited cells into mice. Several weeks later, they harvested the mice and they checked the genotypes. So they were able to see really robust prime editing in the HSCs, which translated to, editing later on from the harvested mouse tissue, up to 40% here. And then the really exciting thing is that you can see a decrease in the sickling phenotype in the prime edited treated cells. So this is just a nice example of using mRNA delivery and electroporation of the prime editing components to execute a therapeutically relevant edit. Another delivery method that has, only been published in the past maybe 2 weeks or so is using dual AAV delivery of prime editing components to install and edit in vivo. So Jesse Davis and Samagia Ben-Skoda in our lab developed a prime editing approach for this dominant negative PCSK9 mutation. And they used this split PE2 max system to deliver the prime editor, pegRNA, and nicking guide to the mouse liver in this dual AAV system. They were able to achieve about 40% editing in vivo in the mouse liver, and this led to a corresponding reduction in LDL produced in the mice. So this is just kind of, if your target is an in vivo target, there are dual AAV prime editing systems that you can use. All right, so we're kind of at the end here to quickly summarize. If you have a new prime edit that you want to do, the optimal prime editing parameters are going to depend on your edit type, the locus, your goal, later down the road cell type, and then you should also be thinking about how you'll deliver these things and what efficiency you need to achieve the outcome that you want. This can all be very overwhelming, but I think the main takeaway from this webinar is that there are concrete guidelines and tools that you can use to make these choices. You don't have to screen every single thing to get, a usable amount of editing efficiency. And I just want to do a few plugs for the lab. So everything I've talked about today is summarized and honestly in greater detail in the Nature Protocols paper that we published, and a lot of really useful constructs are available on AdGene. So in vitro transcription templates, PE max, the AAV constructs I believe are going on there. And then if you want to stay plugged in for future prime editing updates, definitely check out the lab website. And I think David and the lab both tweet a lot about advances in prime editing from our lab and other labs. All right, so with that, I will do acknowledgements and then we'll take questions. First of all, thanks to David. I just finished my PhD in his lab and it was absolutely fabulous. And he's built this amazing technology that we've all been able to work on and contribute to. A huge thanks to Peyton for moderating, and I'll just give a brief background on Peyton because he'll be answering questions too. So Peyton developed with James Nelson ePEG-RNAs that have been really beneficial for prime editing, and Peyton was also involved in the original report of prime editing, so I'm super excited to hear his take on some of the questions that we get later. And Peyton also worked on this protocol paper with me and Alex Sousa, who co-led it, as well as Dr. Peter Chen and Dr. Anahita Vieira. And then I want to shout out a few other members of the lab whose work I summarized today. And kind of my go-to Lou Lab picture is this one of all of us on Halloween. And I will let you try to pick out various people in the picture. And last and most of all, thanks to all the bioprotocol folks for inviting us to talk. This has been a ton of fun, and we really just appreciate the chance to come and chat. So with that, I think we can start doing questions.
Awesome. Yeah. I guess we can just get right into it. The questions have been rolling throughout the whole webinar. So looking through it, we have a couple questions. So I guess I'll start off with this because they're all tying into the sort of same vein of what does prime editing efficiency using other variants of Cas9, PAM variants, other orthologs, how does that compare with regular SP-Cas9?
Yeah, that's a great question. So honestly, it's not great. I think people in our lab have had some luck with SP-Cas9 PAM variants. So like NG, I think does okay. And some of the Cas9 variants published by Shannon Miller and Tina Wang and NBT also do all right. But sorry, I'm trying to get to a slide. In general, like SA-Cas9 does not perform well as a prime editor, which is kind of a shame. But I think one of the things that's important to remember when asking those types of questions, okay, we are here, is that unlike base editing, for example, where you really need a PAM to be in a certain spot in order to do base editing, you have a lot more flexibility with prime editing. So for example, if you're trying to edit this target base, you can do it from either strand. So this protospacer or this protospacer will both work. And your kind of prime editing window is very wide. So for example, if you were trying to do base editing, you'd be restricted to like these like 4 or 5 bases around here. But with prime editing, you can do basically anything downstream of the nick. And you can also get quite far away from your desired edit. And have efficient editing still. So for example, one of the most efficient edits we have is the +26 G to C at the HEC3 locus, which is, very far from the nick, but still works well. So I would honestly, like if I was doing a prime editing experiment and there wasn't a good NGG PAM, I would just try to do something from farther away, or I would use twin prime editing, which can, do changes from even further away. And we didn't talk about twin prime today, but if there are questions about that, we can happily answer them. But I would rather do something like that than use a non-SP Cas9,